All right. So today is going to be day two of Unit 3. Today what we'll be looking at are the organelles and their functions. This will actually be two parts, and I have it split up a little bit differently. Um, but yeah, so today we're going to hit the main structures that are found inside of the cells. Okay. Remember, one thing that you'll have to be able to do is you'll need to be able to identify the structures and know their functions. So just like I said on day one stuff, at the end of the review, I still have those diagrams on there with the answers that follow. This is where you really need to start working once we go over this and you know the functions of them. You really need to start working at being able to label, not only be able to identify them by name, but also label them by uh, label the structures and give their functions to it. Sorry about that. I was being distracted. So the first one we're going to look at is the nucleus. So the nucleus is the control center of the cell. It's kind of like the office. It decides everything that's happening and everything that will happen to the cell. And the reason why it's able to do that is because it contains the DNA. And DNA, of course, makes up your chromosomes. We'll get into that later on as how all that happens, how DNA is genes and chromosomes, and how that all that stuff is really all kind of together, and we'll learn how to break that down. Also, it contains the nucleolus. The nucleolus is this dark spot. If you look at this inside the nucleus, it has this dark circle on the inside of it. That's the nucleolus. I think that's going to be the next slide. It's covered by its own membrane known as the nuclear membrane. That's what covers it right here. You see how it has these little pores here? Well, that's so that RNA can get out. And of course, some things can go in, but DNA cannot leave. And of course, just like the cell membrane, the nuclear membrane, or it can be called the nuclear envelope, controls what enters and leaves the nucleus. So, but it will not allow the DNA to leave the nucleus. Okay? So that's the nucleus. So this right here is about the nucleolus. So of course, this structure on the outside, the big large structure, is the nucleus. This purplish, reddish, magenta structure, this would be the nucleolus. The nucleolus makes RNA and ribosomes. It makes RNA and ribosomes. Now, a lot of students have gotten confused here because ribosomes are made up of RNA, a specific type called rRNA, and they think that, oh, well, that means the nucleolus also makes proteins because that's the job of the ribosomes. No, whenever ribosomes are being made, they are leaving out and they're going into either the rough ER and get caught up in it, or they're going and freely floating in the cytoplasm. They are not doing that in there, or they're not. That structure, the nucleolus, isn't making the proteins. It's just making the ribosomes. So this is the mitochondria. So this mitochondria, of course, this is what's going to be making energy. And a lot of people often think that, well, if the plants have a chloroplast, then animals have the mitochondria. That's not how that works. All the eukaryotes have the mitochondria. Mitochondria is what's going to be actually making cellular energy. It does that through a chemical reaction known as cellular respiration. That's a chemical reaction. And what that means is we're going to break down glucose. Glucose is food, and it releases energy, and that energy is known as ATP. The mitochondria is very unique in the fact that it's kind of bean shaped, and then it has this squiggly line matrix on the inside of it. This actually has a double membrane. And our very last lesson, whenever we're talking about our very last unit, whenever we're talking about evolution, we're actually going to talk about how this is one of the pieces of evidence science has used to sit there and talk about the evolution of cells and how they have changed. Okay, so that's the mitochondria. A lot of you typically know this as the powerhouse or the mighty mitochondria. That's really not how I'll be referring to it, but if that helps you make a connection to this, I don't mind. I just will not be using those terms. So this is the chloroplast. The chloroplast, remember, it's only found in plants. It is going to be capturing solar energy, and that's its job. and that's where photosynthesis happens. So basically, it's going to use light in order to make food. Now, if you notice, this right here, it looks like it's stacks of coins. And we'll get into what all these different things are called because we're actually going to have a unit that is specifically about the chloroplast and the mitochondria. Well, whenever it's doing its job, I mean, if you notice this right here also has two layers of membranes. So whenever we talk about later on, same thing I talked about with the mitochondria, this is going to be another one of those pieces of evidence that science uses. Okay? Ribosomes. Ribosomes, their function is protein synthesis. Now, it's weird. If I ask students what's the function of a ribosome, they'll tell me typically they make proteins. But if I say which organelle undergoes protein synthesis, those same students will look at me like they have no clue what I'm talking about. That's what this word synthesis means. This word synthesis means to make. So proteins are made here. Proteins are synthesized, they're made here. You have two different types. We have freely floating and attached. The freely floating ones would be the ones that we just see, they just look like small dots out in the cytoplasm, while the attached ones are attached to the rough ER. Remember, all cells have ribosomes. So vacuoles, remember though, so the reason why I put these asterisks up here on this top one, about the large central vacuole being plants, that's where the focus will be at. It will be right there. I will not actually ask you about the small sac vacuoles in animal cells, but it is good to already know that they do exist. I don't want you to start going up and go to more difficult levels of biology in the science world and you thinking that animal cells don't have any type of vacuole sacs to contain water, food, and waste. They do. But at this level, as far as knowing the defining characteristics, large central vacuole is found in plants. So you do need to make sure you know the function. It stores food, water, and waste. We could also say, instead of food, we might use words like nutrients instead. And usually it looks like a big swimming pool. It's the big structure. And because of this large structure, it actually pushes the nucleus out to the side. Because typically, like in an animal cell, the nucleus is kind of that center structure. But because of this vacuole, it pushes it off to the side, usually in one of the corners. The lysosome. Remember, this is a defining characteristic for the animal cell. It contains digestive enzymes. So that means these are enzymes that digest, enzymes that break down. They could either break down old cell parts. They're also a recycling center. That way it can, break things apart and try to get the raw parts from it so it could use it again. So that's the job of the lysosomes, animal cells only. A good way of figuring out which one is the lysosomes, typically, so the smallest structure you'll ever see in one's the ribosomes. Those will be the dots. The lysosomes will be a small circle or a small ball, but it won't be nearly as small as the ribosomes. So it's like the next size up, but it'll be one of the smallest structures inside the cell. It'll just look like a circle or a ball if it's a more three-dimensional drawing or rendering of it. Endoplasmic reticulum. So I don't have a picture of it right here. I can go back and show you one in a second. So there are two types. We have the rough and the smooth. The main purpose of it is the highway. And sometimes it's just known as the ER. So the rough, the reason why it's rough is because it has those ribosomes attached to it. And because it has those ribosomes and ribosomes make proteins, rough ER transports proteins. Smooth ER, though, it transports lipids throughout the cell. So just to come over here, typically the rough and smooth ER, the ER, it circles and encapsulates the nucleus. It's usually going around the nucleus. And in our diagrams, you're able to see it more clearly there. And I'll make sure I point that out to you today. So you will need to know the two different types. You'll need to be able to identify the difference between those types like we do on our diagrams. You'll need to know which one transports proteins and which one transports lipids. The Golgi apparatus. Now, a lot of times, you might see Golgi complex, Golgi bodies. These would technically be the Golgi bodies, the small sacs being, the vesicles being pinched off. Anytime you see the word Golgi, don't worry about the second word. Just go with Golgi. A lot of people get this confused with the ER because they both transport things around the cell. But the biggest thing to know about the Golgi is it will modify and package it. That's why we consider it like a UPS, because it boxes it up. So it modifies and packages. If a question is talking about transporting something around the cell and it doesn't use terms like modify or packaging it first or at some point in it, then it's talking about the ER. If it specifically talks about modifying a substance and then packaging it to ship it off, it is definitely talking about the Golgi. So that little stipulation there, that's how you will signify the difference between the ER and the Golgi. All right, so today's review, basically what I did here is I just went through there and I just typed up some different questions or whatever and put them on here. Some of these do go back to day one stuff of talking about the cell membrane and cell wall and that kind of stuff as well, because I just wanted to have a lot of stuff about the structures on here. But so I'll just kind of run through these. If you want to pause these first and see if you can answer them, I'll just run through these and answer them just real quick. I'll give you a second. You can pause it and start answering it yourself, and then unpause it. You can hear the answers. So number one controls what enters and leaves the cell. That's the cell membrane. Number two, made of cellulose, provides structure and support. That's going to be the plant cell wall. Number three, modifies and packages materials for transport. Because it says modifies and packages, I know it's the Golgi. Number four, breaks down old worn out cell parts. That's the lysosomes. Number five, undergoes photosynthesis. That's going to be the chloroplast. Number six, this process makes ATP. That process is known as cellular respiration. Where is ATP made? That's made in the mitochondria. Number eight is a phospholipid bilayer. That's talking about the cell membrane. Over here on the other side, letter A is the cellular highway. That's going to be the ER, endoplasmic reticulum. Makes RNA and ribosomes. That's the nucleolus. The control center of the cell. That's going to be the nucleus. Protein synthesis occurs at the ribosomes. Transports proteins, that's going to be the rough ER. Transports lipids, that's the smooth ER. Where are chromosomes located? Inside the nucleus. Cellular respiration occurs at the mitochondria. And the jelly-like fluid that's inside of the cell, that's going to be the cytoplasm. All right, again, here's your cell. Try to work on diagramming it, labeling it out, and see which ones you're able to actually tell me what they do as well. And you can look back at your notes to find out if you're correct on the functions. Here are your answers for it. If you need to go back so you can pause it and do it, please do that. Remember, on each one of them, I would pause it before looking at the answers. Here's you another cell. Pause it, answer it. About to move on. There's your answers for it. Pause this, check your answers. Remember, try to see if you can write down their functions as well out beside it. This is that next step. This is how we progress in this information. And here's your last one. Pause it, label it out, start seeing if you can put down the functions as well. So I told you I would tell you the difference between show you the difference between that the rough ER and the smooth ER, and this one's really easy to see it on. If you notice the ER, it encapsulates and it goes around the nucleus. The smooth ER, you notice it doesn't have the dots on it, whereas the rough ER does. And a lot of people oftentimes get the Golgi, that's number 7, confused with the ER because they kind of look similar like flattened sacs. Well, if you notice though, the Golgi though isn't connected or isn't right beside the nucleus. So use that to help you out. The ER is right beside the nucleus while the Golgi is off to the side, even though they kind of look similar. Little differences, but they do kind of look similar. All right, and there you go. There's your answers. All right, so that's it for day 2. On day 3, we're going to be picking back up with some more organelles and structures. It'll be pretty short, and then we'll be done because this is the last day.